I'd like to thank Tammy and Sarah for organizing such a wonderful conference and for everyone's interest, as was communicated earlier. I'll be following Kathy's lead and talking a bit about the work that we've been doing over the past 10 years, trying to use neuroimaging with respect to treatment development for addictions. I have a series of disclosures with respect to pharmaceutical.
Just making sure you're sorry.
Can people hear me? Okay.
Oh, here, maybe this kind of is off. That's the issue. There you go.
Okay. Is that better? Okay. So I have a series of disclosures with respect to the pharmaceutical, gambling, and legal industries. And this is not Texas development in addictions, it's treatment development in addictions. And as Kathy was saying, although many behavioral and pharmacotherapies have demonstrated efficacy in the treatment of addictions, we still know that many people do relapse to substance use. And we know particularly little about the neurobiological elements that underlie the active ingredients of the therapies. And we think, like was communicated earlier, that by understanding the neurobiologies linked to these treatments, that we may be able to better optimize treatments and target treatments. So as Kathy described, a major effort has been to try to identify, study neuropredictors and correlates of treatments for addictions. And that a lot of this work has been based in the framework of addictions involving components of bottom-up reward-related drive and top-down executive control. So we've incorporated fMRI tasks into randomized clinical trials that are associated with neurocircuitry underlying motivated behaviors, including assessing reward loss processing and cognitive control, as well as fMRI tasks that are theoretically linked to the proposed active ingredients of the behavioral therapies. And also, as Kathy mentioned in the bridge analogy, I used the Reese's peanut butter cup analogy, that we're bringing two groups of teams together, and each team looks at the elephant from a slightly different perspective. So by bringing the groups together, I think we'll get a fuller understanding of the processes and hopefully be able to implement prevention treatment strategies more effectively. So there's a lot that goes into making the peanut butter cups, and Kathy talked about a lot of the things that we've learned over the past 10 years. I'd also like to say that the groups may come to studying science, studying clinical questions with slightly different viewpoints. And I think, for example, Kathy has done a wonderful job in terms of getting everyone on the PDC team to buy into and recognize the value of imaging people in treatment trials, because not everyone initially was on board with seeing the value or importance to the clinical populations for whom they were trying to provide treatment of getting them to participate in imaging. Then there are also some logistical concerns that we've had to juggle. Kathy mentioned Lucy's limo, but also the idea that people who are running randomized clinical trials understand how important it is to get people from the point of eligibility to randomization really quickly. And that leaves a very limited time slot for trying to schedule fMRI or other brain imaging measures. So that's been something where we've dialogued between the groups and tried to set up a structure that worked well to get a high percentage of people imaged at time one, which tends to be the shortest window. And like Magneto in the first issue of X-Men back in the early 60s, we thought that we might use a big magnet to our best means, not to take over the world in Magneto's case, but our big magnet sort of looks like this. This is a Monet-esque version of our fMRI scanner. And we thought that there were multiple advantages of fMRI with respect to it being non-radioactive. We could engage people coming into randomized clinical trials who were treatment seeking to participate in fMRI studies. FMRI has multiple other advantages, including the task-based effort that aspect that we could then link to the active ingredients of behavioral therapies. Rapid, relatively cost-effective, certainly a lot less expensive than PET, for example, and it does offer high-resolution fine temporal imaging. And as was mentioned, we like to think about the particular behavioral therapies that were being tested in the randomized clinical trials. Think about some of the active ingredients. Those are listed here for cognitive behavioral therapy and contingency management, or at least a subset of the active ingredients. I think Kathy had a much longer list, but for space, these are a few. And we then thought about the potential mechanisms of action from a brain perspective, as well as the brain regions that might be involved, and then thought about tasks that might be particularly well linked to these aspects. And the fMRI tasks are meant to be representative, as there are many other tasks that might be used to tap specific elements. And these are the two that we decided to use going back 10 years ago. One was the standard or traditional historic Stroop cover word interference task, and the other was the monetary incentive delay task. For the sake of time, I'm going to focus on the Stroop and focus on the incongruent versus congruent stimuli presentation. And as Kathy mentioned, our way of thinking about these processes has changed over time. And in our initial forays, we focused on across different studies. We decided to create what we lovingly termed the Frozen 20, which were the first 20 people who we had run through different trials, and then freeze those data and look at the brain imaging measures. And Judson Brewer took the lead on this manuscript, which described using GLM-based brain analytic approaches to link brain activation at treatment onset to urine toxicology-based, self-reported based abstinence, and retention measures. And what we saw was that relatively increased activation within specific brain regions, including regions of the striatum, ventromedial prefrontal cortex, posterior cingulate, were linked to self-reported or urine-based toxicology measures of abstinence, whereas different brain region in dorsolateral prefrontal cortex was linked to treatment retention. And as we heard earlier, it's been really important to think about how brain circuits work rather than brain regions. And there have been a number of different approaches that people have used to understand how brain circuits work. One that we've employed with the help of Godfrey Perlson and Vince Calhoun and others is to use independent component analysis. And independent component analysis, when applied to the same data set of the Frozen 20 plus a control group of 20 individuals, we identified 6 circuits that were linked to, differentially to the activations of incongruent, or the presentations of incongruent versus congruent stimuli. And several of those showed between group difference, the singular opercular and the singular motor circuits showed differential engagement in the cocaine-dependent and healthy control subjects. The medial singular frontal circuit showed differential engagement with respect to treatment retention in the cocaine-dependent group. And then the subcortical and ventral prefrontal striatal brain circuits appeared linked to abstinence measures. And so we, at the time, I think the field was a little less enthusiastic about looking at brain circuits to understand these sorts of questions. It actually took us 7 attempts of submitting this manuscript to get this in press, and we were thankful that it went into the special issue organized by Sarah and Tammy in PAB. Now, we've also used other connectivity-based approaches, and this is a relatively new connectivity-based approach, an intrinsic connectivity distribution approach. And it looks at, in a voxel by voxel fashion, the degree to which any one brain voxel is associated with the activation of other brain voxels and creates a distribution. And what we found was that cocaine-dependent subjects, as compared to healthy control subjects, showed relatively less overall intrinsic connectivity during Stroop performance. However, when you mean adjusted for the degree of overall connectivity, there was a relatively greater intrinsic connectivity within the cocaine-dependent group within these ventral prefrontal and striatal brain regions. And when we looked at, within the cocaine-dependent group, at the relationship with treatment outcome, there was a relationship within midbrain regions, ventral striatal regions, and thalamic regions, such that there was a positive correlation between intrinsic connectivity and percent positive cocaine urine toxicologies. As Kathy mentioned, we've also looked pre-post, and this is in that relatively small, n is equal to 12 sample. So this is not whole brain corrected, but it showed that individuals with mixed substance use disorders, cocaine and other substance use disorders, showed relatively diminished GLM-based Stroop activation post-treatment as compared to pre-treatment. And we had a healthy control group to look at test-retest effects, and some of these activation differences, particularly in the subthalamic nucleus, midbrain areas, were over and above those test-retest effects in the healthy control subjects. But as Kathy mentioned, we've also looked in a larger sample of about 30 plus individuals in the CBT-CM disulfiram study. And we found essentially a similar pattern of results in that post-treatment as compared to pre-treatment, there was relatively diminished activation in GLM-based models in the cocaine-dependent individuals. And also, as Kathy went by really quickly, we wanted to look at how treatment engagement related to changes in functional brain activity, and she showed a prettier version of this slide, trying to copy this over from into a Mac. It seemed to have lost some of the labeling, but the idea being that the changes in brain activation over from pre to post treatment in specific brain regions, including cingulate, pre and post central gyrus, and thalamus, were linked to measures of CBT engagement as well as measures of CM engagement. I also thought it would be good to mention some of the other imaging approaches that we've looked at, and with the hope of integrating these data down the road. These are now looking at that frozen 20 sample, where we found that DTI measures were associated with cocaine abstinence. So poorer white matter integrity was associated with shorter duration of cocaine abstinence, and these DTI measures were relatively diffuse across the brain. And then I think, as Kathy also mentioned, something that's really important is to try to model the data in such ways where we can look to see whether some of the relationships that have been observed in our studies and other studies might work through brain structure and function. So one of the questions that Jin-Sung Zhu, Hedy Koper, and others took the lead on this manuscript, where we looked at pretreatment cocaine use, which was related to treatment outcome with respect to more pretreatment cocaine use linked to greater number of percent negative urine related to more cocaine use as measured by cocaine urine toxicology. We then looked at the extent to which this operated through hippocampal volume. And what we found was that pre-treatment cocaine use, the relationship with treatment outcome was fully mediated by hippocampal volume. So it provides some insight into the relationship, some of the clinical relationships that have been observed and how they may relate to brain structure and function. So I think that this was not meant to be an exhaustive list of the work that we've done. I think Kathy had a spreadsheet of the different manuscripts, and I would direct you to those. But I think that this is meant to highlight the potential of what might be in store for the future. That we're now getting to the stage where we have methods for reliably introducing brain imaging measures into randomized clinical trials, and by carefully measuring the aspects of the behavioral therapies, by carefully assessing brain structure and function, by carefully assessing substance use at different points in time, we should be able to model the data and hopefully get a better understanding of how brain structure and function relates to specific therapies for addictions, and use this to advance clinical care for people. Thank you. And I would be remiss without thanking a range of funding agencies and a range of people, particularly Bruce Rounsaville, who I think really took the lead in initiating these efforts and pulling together a group of people.